A pleasant day to everyone. I am Audrey Cruz, and along with me is my partner, Mr. Matthew Malikdem, third-year environmental biology students from the University of Santo Tomas. With the aid of our co-authors and advisors, we present to you our abstract entitled Stable Isotope Analysis, an emerging tool in connecting taxonomy with ecological interactions among Philippine inland water fauna.
So what is stable isotope analysis? Stable isotope analysis is a well-established method used in elucidating food web structures in aquatic ecosystems. Among the many stable isotopes studied, delta carbon-13 and delta nitrogen-15 isotopes are used in assessing organismal diet. In recent years, stable isotope analysis have contributed in managing conservation strategies by tracing species-trophic interactions and dietary habits. For this abstract, we data mine published primary studies using scientific engines such as Google Scholar and ScienceDirect to be able to consolidate known literature on the utilization of stable isotope analysis in the Philippines along with other Southeast Asian countries. Examples of our keywords are stable isotope fish Philippines and stable isotope fish Southeast Asia. Then we tabulated and organized and analyzed the collated papers through the use of Microsoft Excel.
Upon gathering the scientific papers, we were able to obtain this current status of SIA studies in Southeast Asian inland waters which used carbon and nitrogen stable isotopes. As can be seen in the map of Southeast Asia provided, the Philippines has the highest number of studies with 19 articles, where most studies were conducted in the year 2018. The studies conducted in the Philippines were mostly geared towards Philippine inland water ecology and biodiversity, which contributed to inland water conservation strategies. This includes the study conducted by Briones et al. In 2016 and Garcia et al. In 2014, which used stable isotope biplots to elucidate trophic interactions and feeding habits of species along Lake Sampaloc and Lake Taal respectively. On the other hand, very limited or few to no studies were tallied among the other countries in Southeast Asia. Among the collated articles, an overlap when it comes to the objectives and results were observed, wherein these have also inferred trophic characteristics and diets, although other uses such as distinguishing geographical origins and migrations were also present. Moreover, some studies have also compared morphological and stable isotope data. Given that only a few studies are available to date, this calls other researchers to explore more about the use of SIA.
A common theme can be seen among stable isotope analysis studies in Southeast Asian inland waters. As mentioned, these have shown how stable isotope analysis visualizes trophic interactions among the inland water community, making complex pathways and interactions between pelagic and benthic food webs easier to understand. And this is accomplished through the construction of stable isotope biplot as shown here. Delta carbon-13 and delta nitrogen-15 stable isotope signatures have provided long-term food habits which supported efforts on the organismal gut content analysis by providing a means to identify potential prey items through stable isotope signatures, which otherwise would not have been detected because of variable digestion capacities. These studies then led us to consider that stable isotope analysis as an emerging tool which aids in taxonomy work in the context of ecological applications among Philippine inland water fauna. Since in ecology, we want to learn about the feeding habits, and using this method, we have the means to identify that an organism has been eating a certain food item for a long time, which then brings us to actually identify organisms that are likely part of the diet of another organism, therefore aiding taxonomy work in the context of ecological applications.
And with that, we want to thank everyone for listening, and we hope you all have a good day.